Great. Okay. Hi everyone, my name's Niamh. I am a PhD student in psychiatry, and I'm going to be presenting work today that Shen and I have been working on for the past couple of months. So Shen and I are joint first authors on this paper, and we looked at brain structural associations with depression in a early adolescent sample. Sorry, now I just want to make sure my screen's working. Okay, cool. So yeah, so this is just an outline of today's presentation. So I'm going to just start by giving a brief overview on the background before moving on to the research questions, and then kind of running through methods, results, and discussion. So some background. Well, before I start talking about the paper itself, I just kind of wanted to give a brief background on my research and my kind of PhD. So my research area is adolescent depression, and when I talk about depression in public engagement events and outreach, I often use the metaphor of a whirlwind, and I think this represents the chaos that often accompanies adolescence and the change. So like our bodies are changing, our social world is changing as well. And, it is a time of immense change overall. And I feel that this is pretty apt to describe this year as well for my PhD studies. So for those of you that don't know, so originally, so I started my PhD in 2019, and originally the PhD was meant to be a data collection based PhD. So I was going to look at kind of biological and psychosocial factors associated with risk and resilience for depression. And this involved recruitment this was going to involve recruitment across Edinburgh through schools. So I spent the first year of my PhD preparing an ethics application for the NHS. So that, took up most of my time. And in February earlier this year, we finally got our NHS approval. And about, I think 2 weeks later, COVID-19 kind of broke out in the UK, and there was a complete suspension of face-to-face data collection. So obviously this threw quite a significant spanner in the works, and yeah, so it was a time of immense change for my PhD as well, and it ultimately resulted in me switching to a computational based PhD, because given the fact that data collection was actually suspended for 10 months, the original project wasn't feasible anymore. But I think this has been, obviously quite stressful, but I have learned a lot, and I'm very lucky that, there was resources that I could use to answer my original research questions. So I do hope that, and feel that there are kind of calmer times ahead, and I'm still able to address my original research questions. And the wonderful thing about these big data resources is that there's so much data that you can work with, and neuroimaging research is just an interest of mine in general, and I wasn't able to look at this in the original PhD, and just due to kind of resources and feasibility issues. So it's great that I can look at this now in my adapted PhD. And the overall research question of my PhD remains the same, and that is, what are the biopsychosocial risk and resilience factors associated with adolescent depression? So just a brief overview on adolescent depression. So we know that over half of depression cases emerge by the age of 25. So if we look at the kind of life course as a whole, depression is the time of, adolescence is the time of greatest vulnerability for depression. And unfortunately, depression during adolescence is associated with a more severe illness course. So, a greater number of episodes, as well as, kind of a more severe, symptom presentation. And, this can lead to a propagation of difficulties across the lifespan. So we know that depression during adolescence is associated with lower educational attainment, as well as strained social relationships. And this really does compound the burden of depression during what is already a time of significant challenge. So when we assess depression in, and kind of psychopathology in general in young people, we use a multiple informant approach in the diagnosis. So this typically involves a caregiver report as well as a child self-report, and then sometimes a teacher report is also incorporated into it. And this gives the kind of, this is the gold standard, and the aim is that it gives us a more kind of holistic understanding of the difficulties that the young person is experiencing. But kind of one of the challenges of multiple informant, the multiple informant approach, is that, some discrepancies do arise between reports. And this has been documented in the literature, that there can be discrepancies between caregiver and child report. And because some would argue that, can we rely on a parent's outsider or objective interpretation of the child's internal state, and then during adolescence, a young person, it's a time of significant development, neurodevelopment in particular. So can a child accurately perceive and report on their internal state? So given the fact that depression is quite common in this developmental period, it has led to a lot of research in this area. However, there has been a paucity of research on the kind of neurobiological features of depression in adolescents compared to adults. So we know that there are alterations in brain structure associated with depression, and this is from kind of a wealth of literature on adult depression. So over the past 30 years, the advent of large-scale neuroimaging studies, such as the Enigma Consortium, amongst others, has really given us unparalleled insight into the kind of brain features of depression. And this work has kind of revealed some findings, including reduced hippocampal volumes associated with major depressive disorder, as well as reduced cortical thickness in frontal areas. And then we also see white matter alterations in kind of tracts connecting different parts of the brain. So in frontolimbic tracts as well as frontothalamic tracts. So, it's evident that there are widespread structural alterations associated with depression, but the vast majority of these studies have been done in adults. And while there have been some efforts to understand the kind of imaging features associated with adolescent depression, these have been few in number. And of those that do exist, there are kind of discrepancies in terms of the regions involved, as well as in effect sizes, and this is likely due to the small sample sizes in these studies. There is a notable exception. So within the Enigma Consortium, there is a sample of adolescents. So this sample has reported lower total surface area, as well as regional reductions in frontal areas. So some of these are similar to what we see in the adult literature, but then there are notable differences, such as the lower surface area, and I'll talk about this later on in the talk. But the thing about the Enigma adolescent sample is that it's so smaller, relatively smaller compared to the adult sample, and then also it's focused on older adolescents. So, given the fact that adolescence is the time of greatest vulnerability for depression, and also the fact that adolescence is a time of significant neurobiological change in the brain, this gives us kind of reason to, if we really want to understand the origin and development of depression, then we really need to trace the roots of depression in the developing brain. And this is something that we hope to do in our current work. So this leads me on to our research question. So our primary research question was to investigate what cortical structures and white matter microstructure are associated with depression in adolescents. And then also, given the discrepancy in caregiver and child report, we also wanted to look at the association between this reported discrepancy and brain structural associations. So moving on to the methods. So just kind of, I first kind of wanted just to have a quick chat about the Adolescent Brain Cognitive Development Study, so the ABCD study. So for those of you that haven't heard of this study, it's absolutely wonderful. It's been such a pleasure to kind of get to know the data set and to learn more about this study. And honestly, it really is kind of a monumental development, and it's a kind of monumental development in terms of neuroimaging research in adolescents and young people. And, you know, it's hoped that the findings that emerge from the study will really advance our understanding of how the adolescent brain develops. So this is a kind of large-scale population-based study in the US. So it started in 2016. And I think baseline recruitment was from 2016 to 2018. And in the baseline recruitment, they recruited over nearly 11,000 young people aged 9 to 11 across the US. So even though it's kind of so it's from 21 sites across the US. So it is a population-based study. The kind of study designers are reluctant to use the phrase representative because, you know, it's not entirely representative of the US population as a whole, but they did try to make it as close to that as possible. And it includes a host of measures, including imaging data, mental health assessments, and then a range of environmental and cultural measures for all participants. So at the time of writing this paper, only the cross-sectional data was available. So that was from wave 1. But since then, I think it was in late November, they released the second wave. So there's now a full follow-up data set for all of the kind of behavioral and mental health assessments. And then there's half of the follow-up imaging data. So I think the full wave of imaging data will be released next year. But for those of you who aren't that familiar with the ABCD study, I'd highly recommend checking out their website. So it really is, great, like a fantastic resource. So moving on to the methods we used in the current study. So just kind of in honor of full transparency. So this is the paper that Shen and I co-led. So Shen led the data preparation and analysis, just to highlight that. So in terms of the participants that we used in this study, so we used the annual data release 2.0.1, and we used an unrelated sample for this study, and this resulted in a final N of nearly 10,000 young people. And these were aged 9 to 11, and around 52% were male. In terms of the brain measures that we looked at, so FreeSurfer and the Desiccant Atlas was used to generate the cortical measures. So the cortical measures that we used were surface area, cortical thickness, volume, and sulcal depth. And then the white matter measures that we used were fractional anisotropy and mean diffusivity. And for those of you that aren't too familiar with these measures, so FA and MD are kind of a measure of the integrity of these white matter tracks that run across the brain. So lower FA would be indicative of kind of reduced integrity of these tracks. And we looked at both global and regional measures. In terms of the mental health assessments that we looked at, so depression was quantified using the KSADS, which is kind of a standardized, well-established metric used for quantifying psychopathology in young people. And we used a binary and a continuous measure of depression. So the binary measure was either a diagnosis of major depressive disorder and a control. And then we also kind of wanted to look at depressive symptoms more broadly. So we used a continuous measure, and this was derived according to the DSM, and it kind of consisted of mild, moderate, and severe depressive symptoms. We had caregiver and child reports of these symptoms, and we also included environmental and cultural factors. So then in terms of the statistical models, so we used general linear models and linear mixed effects models in our analysis, and included covariates, and all of this was conducted in R, and the covariates we included are here. And just kind of to highlight that we included parental mood as a covariate, as well as the kind of imaging covariates, and we applied FDR correction for multiple comparisons. And then we also conducted additional sensitivity analysis to control for site effects and scanner effects, and then kind of just to highlight in particular is we also controlled for youth antidepressant effects. Even though there weren't many young people on antidepressants, it was important to control for medication. So moving on to results. So just to start off with some sample characteristics. So you can see here that, even though like depression was the focus of the paper, they're overall, most of these young people kind of have navigated adolescence or the early stages of adolescence without developing depressive symptoms. And I think that's something to keep in mind, because a lot of the time, even I kind of have to remind myself that, there are a significant number of young people that will remain well during adolescence. And while it's important that we investigate the factors that influence why young people, or why some young people don't remain well, it's always important to highlight that most people do remain well. So here you can see that there's the major depressive disorder cases and then the depressive symptoms. And you can see that, there are kind of, for 9 and 10-year-olds and 11-year-olds, there are quite, a relatively significant number of cases reported within the sample. And maybe that's something that we could have a chat about afterwards. But one thing that we noticed while kind of doing this analysis was that there were kind of more, a higher diagnosis rate within the sample compared to kind of European samples. And maybe that's something to do with the US population, but it's just something to keep in mind regardless. So, okay, so firstly, starting off with global brain measures. So you can see here on this figure in panel A, we have the depressive symptoms and MDD reported by caregivers. And then in panel B, we have the child reports. And, so first of all, looking at depressive symptoms for caregivers and MDD, we can see that depressive symptoms are associated with reduced cortical volume as well as surface area. And so in terms of the cortical measures, and then for the white matter measures, we see reduced FA as well. And these findings so we see similar findings for the depressive symptoms reported by children. So so in terms of the global measure, or the global brain measures, these findings are similar to that which we see in the adult literature. So in the adult literature, the research has demonstrated that there are lower cortical volumes associated with depression as well as lower FA. And as I'll discuss later on, we do see this kind of notable difference between the adult literature and the adolescent literature in terms of the reduced surface area. So that's something that we don't see in adult MDD. In fact, we see a reduction in cortical thickness, which isn't present in the current sample of adolescents. So moving on to the regional brain measures. So you can see here in this figure, these are p-value plots for the association between depressive symptoms and the different cortical and white matter microstructure measures that we looked at. And just in the interest of time, I've only shown the depressive symptoms here and not the MDD. There were more significant associations for depressive symptoms compared to MDD, but there were still similar findings between the two measures. You can see here that in panel A, we have the caregiver report, and then in panel B, we have the reports from the child. So firstly, looking at the cortical measures. So we can see that there's reduced kind of cortical volume in frontal and temporal regions, as well as reduced cortical surface area in these regions as well. And then in terms of white matter measures, we see reduced FA in the kind of frontal limbic tracts. So you can see here that in the kind of superior longitudinal fasciculus and the superior cortical striate, we can see reduced FA there. So this would suggest that there is kind of altered integrity in these tracts. Okay, so then in terms of our reported discrepancy, so actually, sorry, before moving on to this, so one of the kind of main findings that that we had in the study is that we saw more numerous and more significant associations between caregiver reports of child depression and brain associations. So that was kind of something that was quite interesting for us, and like what is driving this kind of association. So we then as kind of exploratory analysis, we looked at the factors that are influencing this discrepancy. So in this figure here, we can see the factors associated with more reported discrepancy. So we looked at some socio-environmental factors that are included within the ABCD study. So you can see here on the right-hand side, these are the list of the kind of external factors, and the ones with an asterisk are the parent report. So and this is the absolute discrepancy between caregiver and child reports of depressive symptoms. So you can see here that kind of, factors that, would suggest like poor social cohesion and kind of, family conflict, such as family conflict, sleep disturbance, neighborhood safety are associated with like more reported discrepancy, while factors that would suggest greater social cohesion and kind of, more pro-social behavior within the house or within the household and within the kind of neighborhood are associated with less reported discrepancy. And, these findings aren't exactly surprising that, greater social cohesion is associated with reports that are more aligned. So we can chat about this in the discussion, but there's certainly a lot to unpick here. Okay, so kind of the first main finding of our study was that we replicated findings in adult MDD literature. So we found that in the sample that major depressive disorder and depressive symptoms are associated with reduced global cortical volume and global FA. And then we also see, in terms of kind of regional measures, we see reduced white matter integrity in these frontal limbic circuits. And this would suggest, so the fact that we see an alignment between the adolescent literature and the adult literature suggests that these brain structure alterations may be present very early in the stages of depression. So the fact that, we see them, in these 9 and 10-year-olds, they're in the really early stages of adolescence, and then, it suggests that these alterations persist across adolescence and into adulthood. And, in terms of the kind of reduced integrity in these white matter tracts in the frontal limbic circuits, research has suggested that the reduced integrity in these association fibers is related to compromised cognitive control, and that, it's these alterations that may underpin the clinical features that we see in depression, such as, the kind of maladaptive thinking styles and coping strategies. So so that's quite interesting there, that we do see the kind of a parallel between the two age samples. And then in terms of the adolescent specific findings, we do see some notable differences. So in this sample, we saw global and regional reductions in cortical surface area. And this is quite a, departure from what we see in the adult studies. So in adult studies, we often see reduced cortical thickness, and this is something that we didn't see in the current sample. And this, raises a lot of interesting questions, because firstly, our findings were in line with those of Enigma. So the Enigma sample that looked at, or the Enigma adolescent sample also found these kind of regional and global surface area deficits. And this is even in an older sample of adolescents. So the fact that these are present in the early stages of adolescence, of adolescent depression, suggests that maybe these, cortical surface area alterations are related to early life risk factors specific to depression. So, for example, in the literature looking at childhood trauma and low birth weight, we also see these surface area deficits. And I suppose there's quite a lot to unpick when we kind of, when looking at these findings, because, why don't we see these surface area reductions in in the adult MDD literature? Is it that, do they not disappear over time, but do they change over time? And why do we see cortical thickness in the adult MDD literature, but not in the adolescent depression literature? And there's been quite a lot of debate around this. And some of the kind of Enigma authors would suggest that perhaps these cortical surface area reductions actually represent kind of a general, like, kind of vulnerability to mental illness, and that perhaps the reason that we don't see them in the adult MDD is that these adolescents actually go on to develop other mental health conditions, such as schizophrenia or bipolar disorder. So maybe that's why we don't see them later on. So yeah, so there's quite a lot to discuss there. And I think, as the ABCD sample grow older, we'll be able to probe these associations further, because as I said, this was just a cross-sectional sample that we used in the current study. So moving on to reported discrepancy. So as I said, we did observe some similarities between global cortical volume and surface area. But overall, caregiver report demonstrated stronger and more numerous associations with imaging features. And then when we looked at the kind of factors that are influencing the discrepancy, we found that, there are these contextual factors that do seem to be driving the discrepancies. And, as I said, the integration of reports may pose difficulties in the diagnosis of depression, because, can a caregiver accurately report on the child's inner kind of mental state, and then does the young person have the ability to can accurately report on their inner state? And we know that this is kind of a neurodevelopmentally sensitive period. So yeah, so that's kind of, something to consider there. And then one, I suppose kind of one of the main takeaways from the findings on reported discrepancy is that, we see, first of all, we see stronger associations between caregiver reports and adolescent depression. And then we also see, so on one hand we have that, and then on the other hand we see that there are these environmental factors that are influencing the kind of degree of discrepancy. So it may be that there are these kind of contextual associations kind of with things like sleep disturbance and family conflict that are actually driving the association that we see between caregiver reported depression and these brain structures. So this is something that we're going to kind of look at further as a follow-on from this study. So kind of going back to the research questions that I kind of brought up at the start of my talk. So we wanted to see what were the cortical structures and white matter microstructure associated with depression in adolescents. And as discussed, we see similar findings to the adult literature, but then also in terms of reduced cortical volume and reduced white matter integrity. Then we also see notable differences in terms of the reduced surface area. And then, as I mentioned, we see we see that caregiver reports of depression have stronger associations with adolescent depression. So although this sample, or this study benefits from a very large sample, there are certainly limitations. So as I said, it was a cross-sectional sample, and once we get the full imaging sample from wave 2, we'll be able to look at the longitudinal longitudinal effects to see whether or not there are any causal effects or causal associations. And also, I suppose, to look at whether or not the reported discrepancy persists throughout adolescence, and do they become less or more aligned as the young people get older. And then also in terms of the heterogeneity of depressive symptoms. So although we had a binary and a continuous measure of depression, we didn't look at subtypes of depression. And there's quite a lot of talk around, the heterogeneity of depression, that we really need to kind of expand our definition of what depression is. So, including subtypes of depression, as well as subclinical symptoms, because we know that like a third of adolescents who experience subclinical symptoms during adolescence will go on to develop major depressive disorder as an adult. So, kind of having a wider definition of depression might be able to kind of, further our understanding of the neurobiological basis of it. And then also, this is a very young sample of adolescents. So they're really at the early stages of their teenage years. And then, with the onset of puberty will kind of, bring about a host of other biological changes. So that's actually something that I'm going to look at, or that I'm looking at in my next project, is looking at the association between puberty, brain structure, and depression. So yeah, I'm not sure how we're doing on time. Okay, I think we're doing okay. So just kind of to talk about that briefly. So I'm in the process of doing a puberty registered report on pubertal timing, imaging features, and depression using the ABCD sample. So for those of you who aren't too familiar with the registered report, so this is a kind of open science practice and a kind of a different way of publishing research. So normally when we kind of publish a research article, the peer review is all at the end. But in a registered report, the peer review is split into 2 stages. So you essentially write your introduction and your methods, which includes a very detailed data analysis plan. And then this is sent off for peer review. And if all going well, you'll get an in principle acceptance. And this means that regardless of what you find in your study, that the journal will publish your results. And this is a way to kind of, prevent p-hacking or prevent bias in science, and that you're kind of pre-specifying all the analysis you're doing, and then you're kind of testing those hypotheses in your analysis rather than hypothesizing after the results are known. So this has been something that I've been working on for the past couple of months. And although, it has been quite a lot of work, I think, for me, it's been a really good exercise just to properly think about what you're doing in your analysis plan and why. And essentially, you have this kind of blueprint now that you just follow when you do your analysis, which has been great, even though, you are kind of front loading your work. I think it definitely pays off. So the question that I'm going to look at in this registered report is, does brain structure mediate the association between early pubertal timing, so this is pubertal development relative to your peers, and depression during adolescence? So just briefly, this is my mediation model. So I'm going to be looking at whether or not imaging features mediate the association between pubertal timing and depression. So we know that early pubertal timing is associated with an increased risk for depression. And then kind of, from the study that I've just discussed, we know that there are associations between depression and imaging features, but I'm going to look at whether or not does kind of brain structure, mediate this relationship that we see. And because we now have follow-up data in the ABCD study, I'm going to be using the depression from time point 2. So these are going to be 11 and 13-year-olds, and then imaging and pubertal timing measures from time point 1. So so I think that's it. I just wanted to say, kind of a huge thanks to Shen, who's kind of led this work with me, and then all of our co-authors as well. And yeah, thanks very much, and I welcome lots of questions.